Hello students, so in this video I'll be discussing about the structure of DNA. DNA, as you know, deoxyribonucleic acid is the genetic material of all living organisms, right from bacteria up to human beings. So all of us, we have our genetic material as DNA. DNA codes for all the kind of proteins which will be synthesized in our body, and that will decide how we will look like, how our cells will function and everything. So before going into the structure of DNA, let us have a look actually how the DNA molecule is packed. If you look into this figure, you'll find something is written here that an human DNA molecule is around 6 feet in length. So you can imagine 6 feet is such a long length compared to the microscopic nature of the nucleus inside which that DNA needs to be packed. So inside nucleus we have chromosome. Chromosomes are actually rod-like structure. So in order to understand chromosome, let us do an analogy. You must have seen some woolen balls. So in market, when you see woolen balls, you'll find that the woolen balls, they look like this. The wool strand is folded and folded and it forms a rod-like structure. So same is the case with DNA. So chromosomes are highly supercoiled actually DNA molecule. Chromosomes do not have any membrane just like cell organelles. So if we magnify a portion and we magnify it even further, we'll find that it is a kind of supercoiled structure, something like that of a telephone wire. And this supercoiling is made up of some beaded structure. Each bead, if we magnify and see, we'll find that there are some globular structure, ball-like structures around which some wire-like structure is wound around. So that wire-like structure is the DNA molecule and this ball-like structure are the histone proteins. So these histone proteins actually help in coiling or in packing of the DNA molecule around it. So if you remove the histone proteins, you will find that that wire-like structure on being magnified, it is this DNA, double stranded DNA molecule. So that is the overall idea of DNA molecule. Now this DNA molecule is a complex biomolecule. So it will be made up of different simpler components. So what are the components of DNA molecule? Let us have a look. First we'll have a look at the chemical structures, then we will see the theoretical portion. So if you look here, just have the figures for you. So the first, so let us think that DNA is a dish and we are trying to cook up DNA. For that, for cooking dish, you need different raw materials. So the first raw material of DNA is the sugar molecule. So the full form of DNA is what? Deoxyribonucleic acid. So that deoxyribo part comes from this deoxyribo sugar. So if you look here, there are two sugar molecules here, deoxyribose and ribose. These are pentose sugar. Why pentose? Because these are 5 carbon sugar. First, second, third, fourth carbon, they are present in the chain and the fifth carbon is outside the chain. What is the difference between deoxyribose and ribose sugar? In the second carbon position, in deoxyribose, there is no oxygen, and in case of ribose, there is oxygen. So that's the basic difference between deoxyribose and ribose in the second prime position. This 2 dash is also called 2 prime position. Is that clear? Okay, so that's the sugar molecule. So in DNA, we'll have deoxyribose. In RNA, we have ribose sugar. After that, we need nitrogenous bases. Now, nitrogenous bases are of two types. One is called purines, another one is called pyrimidines. Now, purines are double ring structure. Adenine and guanine are the purines which are present. So what are the double ring? You simply remember it as one hexagonal ring and one pentagonal ring. Now this is imidazole ring, but easy way of remembering is this way. One is hexagonal, one is pentagonal ring. These are heterocyclic compounds. Why heterocyclic? Because the entire ring is not made up of carbon. There is carbon and nitrogen. So there is adenine, there is guanine. These are purines. And pyrimidines. Pyrimidines which are present in DNA is cytosine and thymine. These are hexagonal ring heterocyclic compounds. Okay. So this thymine is exclusively found in case of DNA, and in case of RNA molecule, instead of thymine, you will be having uracil. So these are the pyrimidines. Is that clear? So that's the second component. And the third component is a phosphate group. This phosphate group is derived from phosphoric acid, PO4 3 negative, and it gives the characteristic acidic nature to the DNA molecule. So these are the three basic raw materials that will be required: the sugar, the nitrogenous base, and your phosphate group. Now initially what happens? Initially the sugar will combine with the nitrogenous base to give you what is called your nucleoside. So let us have a look here. So you see here, this is the sugar, this is the nitrogenous base. So this is deoxyribose sugar combining with thymine. So what is the name of the nucleoside? Deoxythymidine. Here this is the deoxyribose sugar combining with cytosine forming deoxycytidine. Now in case of deoxyribose sugar, if there is ribose sugar, we don't have this deoxy part written, it is simply written as cytidine. Similarly, deoxyribose combining with adenine, it forms deoxyadenosine. This is the purine which is combining. These two were pyrimidines. And another purine which can combine is deoxyribose sugar with guanine. So that will give you deoxyguanosine. So these are the four nucleosides which goes into forming the DNA, and these are the name of the four nucleosides which will go into making of RNA: uridine, cytidine, adenosine, and guanosine. So these three nucleic acid, the nitrogenous base are common, that is your cytosine, adenine, and guanine. These are common for RNA and DNA, but in case of DNA you get thymine, and in case of RNA it is uracil. So that is the difference. Now this nucleoside will then combine with the phosphate group in order to form what is called nucleotide. So let us have a look at nucleotide. So this is how a nucleotide is formed. So you have pyrimidine nucleotide and you have purine nucleotide. So for example, these are all being shown with the help of ribose sugar. Let's come to one, your thymidine monophosphate, deoxythymidine monophosphate. So that is the kind of nucleotide you find in DNA. Like that, three other nucleotides will be there. What is here? This is the ribose sugar. Then this is your thymine. So this is deoxythymidine, that is the nucleoside. When it combines with the phosphate group, it forms deoxythymidine monophosphate, because one phosphate group is that, DTMP. So similarly, so now let us have a look at the theory of what are the different nucleotides which can be formed. So these are the pentose sugar is deoxyribose sugar, nitrogenous base. We have, just make it slightly clearer. Okay. So purines is guanine, adenine, pyrimidine is cytosine, thymine for DNA, uracil for RNA, phosphate group is there. Sugar and nitrogenous base combines to form nucleoside. So deoxyribose plus guanine gives you deoxyguanosine. Deoxyribose plus adenine gives you deoxyadenosine. Then deoxyribose plus cytosine gives you deoxycytidine. Deoxyribose plus thymine gives you deoxythymidine, which is there. This one is exclusively in case of DNA, and these are the nucleoside which you will find in RNA. So these are so now what I told you, that nucleoside, they combine with phosphate group to form the respective nucleotides, and nucleotides will join to form DNA. So deoxyguanosine plus phosphate group, it gives you deoxyguanosine monophosphate, DGMP written in short, or it is also called deoxyguanylic acid. Deoxyadenosine plus phosphate, it gives you deoxyadenosine monophosphate, DAMP, और deoxyadenylic acid. इससे कुछ याद आया? एडिनोसाइन मोनोफॉस्फेट, एडिनोसाइन डाइफॉस्फेट, যদি তিনটা গ্রুপ লগ লাগি যায়, if 3 phosphate groups gets attached, this will then become adenosine triphosphate, ATP, that is an energy rich compound. Here it is only one phosphate group. Deoxycytidine plus phosphate, it is deoxycytidine monophosphate, DCMP or deoxycytidylic acid, and deoxythymidine plus phosphate, deoxythymidine monophosphate or deoxythymidylic acid. These are the nucleotides which will go into formation of DNA, and these are for RNA. Is that clear? So now we will, so that was the theory part. Now we'll come back to how the actually the nucleotides join with each other. The nitrogenous base there join with each other. So you should always remember one thing, that guanosine always pairs with cytosine with the help of 3 hydrogen bonds, and adenine will always pair with thymine with the help of 2 hydrogen bonds. So that is how the bonding between nitrogenous base takes place. Guanine will never pair with thymine, cytosine will never pair with adenine. So CG is the pairing or AT is the pairing. That kind of pairing we will see when the nitrogenous bases will combine in forming the DNA chain. Now going to the DNA chain, how do the DNA molecules there join? So you see here, if I enlarge it, bring it down. Okay. So let's observe this portion. So you see, this is the sugar molecule, this is the nitrogenous base, and this is the phosphate group. That means this makes one nucleotide, correct? Then this is one sugar molecule, this is one nitrogenous base, and this is one phosphate group. So this is another nucleotide. So how the two nucleotides will join? They will join with the help of at this particular position. So the phosphate group of one nucleotide will combine with the other nucleotide at the third carbon position. This is the first carbon, second carbon, third carbon. So at the third carbon position, the phosphate group of the one nucleotide will combine with the carbon of the sugar group, third carbon of the sugar group of the next nucleotide with the help of a bond, and that bond is called the phosphodiester bond. So that is how the chain is created. Now similarly, here also this is one nucleotide, this is the sugar, this is the nitrogenous base, and this is the phosphate group of this particular nucleotide. This is the sugar, this is the nitrogenous base, and this is the phosphate group. But if you look here, the difference, the nucleotide are in a reverse position, something when they will join something like this. So that means here the sugar molecule is like this, here it is inverted. I'll just show you in a simplified way. So that creates polarity in the difference of polarity in the chain of the DNA molecule. I'll come back to this structure once again, but before that let me explain to you. For example, suppose this is a sugar molecule, this is the nitrogenous base attached to the first carbon, this is the first, second, third, fourth, and the fifth carbon is outside, and with the fifth carbon the phosphate group is attached. So this is one nucleotide. When the next nucleotide will join with this, where it will join? At the third carbon position. So at the third carbon position we have the phosphate group of the next nucleotide. Here will be the 5 carbon of that nucleotide, the sugar molecule. This is the sugar molecule, this is the first carbon, second, third, fourth, and fifth carbon, and in the first carbon nitrogenous base is attached. So this is how the chain will keep on elongating. Is that clear? So अभी क्या होता है देखो कि suppose this is one chain of the DNA molecule, the last nucleotide which will be on this side, इसका third carbon position, the 3' carbon will be on this side. So this is called the 3' or the 3' end, and the last nucleotide which will be on this side, that will have which carbon? The 5th carbon on this side. So this is called the 5' or the 5' end. So the polarity of this chain is 5', 3' polarity. Here on the other side what happens? This is the nitrogenous base. It is also attached with the sugar molecule, but the sugar molecule is reverse, I told you. So first, second, third, fourth, and the fifth carbon is outside. इसका ये phosphate group है अपना. So this is one your nucleotide. Next nucleotide, suppose this is the next nucleotide, nitrogenous base. So this is the sugar molecule. First carbon, second carbon, third carbon, fourth carbon, and the fifth carbon will be outside, and this is the phosphate group of this particular nucleotide. And this nucleotide will join with this nucleotide, where with the help of this phosphate group in the कार्बन पोजीशन. सो फॉर दिस पर्टिकुलर चेन, ये चेन में इस तरफ का जो लास्ट न्यूक्लियोटाइड होगा, द थर्ड कार्बन इज ऑन दिस साइड. सो this will be the 3' end, and the last nucleotide which will be on this side, it will have which carbon on this side of the sugar molecule? The 5'. So this will be the 5' or the 5' end. So this will have a 3' 5' polarity. So this is the how the DNA chains are formed, the two chains are formed. और बीच में क्या रहता है? In between you have the nitrogenous bases. If it is adenine and if this is thymine, I told you adenine always pairs with thymine, they'll pair with each other with the help of 2 hydrogen bond, and if it is cytosine and guanine, they'll pair with each other with 3 hydrogen bonds. Now 2 purines and 2 pyrimidines will never pair up. Why? Because suppose 2 pyrimidines pair up, here the DNA molecule, the breadth of the DNA molecule will become less. And if suppose 2 purines they pair up, what will be the problem? Here the DNA molecule will become very broad. But DNA molecule, you should remember, it is a genetic material. It needs to be as stable as possible. So it needs to have a uniform thickness, and that uniform thickness is achieved by pairing of one pyrimidine with one purine, so that overall there are always 3 rings in this central part and it maintains a uniform thickness. And there is another rule here which is called the Sharjaf's rule. What is the Sharjaf rule? Is that the ratio of total amount of purine by the ratio of total amount of pyrimidine is always 1:1. It has to be, suppose purine, what are the 2 purines involved? The purines are, one is adenine and another one is guanine. And what are the pyrimidines involved? Pyrimidines involved are, one is thymine, another one is cytosine. So total amount of purine will be equal to total number of pyrimidine. It has to be, because one purine always adds with one pyrimidine, whatever. So its ratio will always be 1:1. This is called Sharjaf's rule. So that is the overall structure of DNA molecule. Now let us go back to the diagrams that I have downloaded. So you see here. So let's come back to this. So what we can actually see here is that in the diagram you can see that this is the one DNA chain, this is another DNA chain. The central part is made up by the nitrogenous bases, guanine pairing with cytosine with the help of 3 hydrogen bond, adenine pairing with thymine with the help of 2 hydrogen bonds. And if you look at the DNA molecule, you see here. Now going to the macro level or the morphology of DNA molecule, when the DNA makes this coil, the double helix, one pitch, the length of one pitch means one full round, one single round of the DNA molecule, it is of 34 angstrom length, and within this one pitch there are 10 such cross bridges of the nitrogenous bases. So the approximate distance between the two nitrogenous bases is 3.4 angstrom, and the diameter of the DNA strand is 20 angstrom. Another thing, I forgot to tell you about this. Since this strand is 5'3' and the other strand will be 3'5' polarity, that's why the DNA strands are called anti-parallel. They are parallel, no doubt, but they are in opposite polarity, that's why the term anti-parallel. So I hope the structure of the DNA molecule is clear to you. In the next video I will be discussing about the replication of DNA.